keep you entertained for about 45 minutes on my topic. It's about early evolution, okay? And this is a problem, all right? Why early evolution? And it's even going back to the origin of life. All right. Now, the origin of life is a horrible topic. First of all, it's not really science, because even if we could make a reactor where we put chemicals in on one side and out comes E. coli with the wrong genetic code on the other, we still couldn't prove that our ancestors arose that way. We would just have a narrative that would make it more plausible. Okay, so it's not really science. Nonetheless, people want to know about it. Right? The society wants to know, how is it possible? But I'm not doing this for society. I want to know, okay? How is it possible? And so we have the great honor of getting research grants to investigate this stuff. And then I was telling the students earlier, the only obligation we have is to tell society in the written record on a regular basis what we have done with their money. And so, this is what I'm doing today, trying to explain what we're doing with your money. And so that needs to be said. Also, I'm going to give you an hypothesis for the origin of life, and you may have everybody has their own ideas about this. And so much of what you hear today may be different from what you believe, and I am not interested in trying to convince anybody about what I think, okay? Everybody should believe what they want to believe. And furthermore, it doesn't make any difference whatsoever whether what the hypothesis that I present to you is true or not. I already have a permanent position. This is very important, right? So this gives me a little bit of freedom to think about these things. All right, so given that, this is it's an exercise in evolution. Evolution starts right here. Excuse me, I've got a little bit of something in my throat, a cough, and so I'm going to be clearing my throat a lot today. Please forgive me. This is a segment of the only figure in Origin of Species, and it shows, it shows Darwin's principles, natural variation and natural selection. Okay, so he didn't have a mechanism for natural variation. He just observed that it occurs because he went to dog breeding shows and pigeon breeding shows, and he noticed that the progeny at each generation were not identical. So there was some mechanism generating variation. And then his big idea, natural selection, some of the progeny are more fit to make it to the next generation than others. And using these two principles, so Darwin, we can explain all of the evolution all of the biodiversity in the natural world. It's still basically true with two minor modifications that Darwin was not concerned about. They occur in microbes. The first is endosymbiosis. Today we know that plastids and mitochondria come from free-living endosymbionts, cyanobacteria and proteobacteria that took up residence within a host cell to give rise to eukaryotes and plants. So we had mergers of preformed cells giving rise to novel taxa at the highest level. Darwin had nothing like that in mind, but that's specific to microbes, and he wasn't concerned about microbes. And the other mechanism that we know that runs counter to this principle is lateral gene transfer, also specific to microbes, and Darwin was not concerned about microbes. We know this from lateral gene transfer is real. We know this from hospitals with antibiotic resistance genes being passed around. It's a real problem. So, but what is true also is that when it comes to the origin of life, Darwin's theory works much better if you already have something that is alive. This transition here doesn't really work with his principles, and there we have to there has to be something chemical going on, because if you don't have genes, if you don't have heredity, how do you get to genes and heredity from chemicals? And so that's what we'll be dealing with today, that problem, that early phase of evolution. This is just a reminder. Many of you have heard of the RNA world. Yeah, you've heard of the RNA world. And the RNA world presumes that bases were there in sort of like a prebiotic soup. But this is just to remind us that the bases today are made from smaller molecular weight compounds, many of them relevant for biology, whether it's tetrahydrofolate or glutamine or aspartate, CO2, glycine, aspartate, and carbamyl phosphate here from ammonium, made from ammonium CO2. It's also interesting to note that, excuse me, even in human de novo purine and pyrimidine biosynthesis, there is a carbon atom in each base that comes from CO2. And so there's a class of theories called autotrophic origins that say that life started out from CO2. If that's true, then there is a trace of autotrophic origins in every base. Another thing to mention is that I'm a biologist, and I look at the world very much in terms of energy metabolism, core physiology, the processes that keep us alive. Life is a chemical reaction. If you don't believe me, take a plastic bag, put it over your head, and hold it real tight, and then we'll continue the discussion. After 53 seconds, life is a chemical reaction. For us, it's the burning of sugars, carbohydrates, fats, and amino acids in our mitochondria to generate energy. For all cells, that's basically the process. We have some core exergonic energy-releasing reaction at the center of metabolism that generates ATP and all else. And it's very interesting to note that if we look around among modern cells, there are actually only two ways to make ATP. There are fermentations, and these are substrate-level phosphorylation involving the disproportionation reactions of reduced organic compounds. And these are always derived from these kinds of organisms here that generate an ion gradient, either phylogenetically derived or derived ontogenetically, because fermenters always need large amounts of chemically homogeneous substrates that they can ferment. Okay, they can't work on, for example, stardust, organics from space, which are much too heterogeneous. They consist of a very complex mixture of ppm and ppb concentrations of various isomers of carbon compounds and polyaromatic hydrocarbons. So fermentations are actually derived from this. And this is the other way to make ATP. It's basically the universal way. It was discovered by, thought up by this guy named Peter Mitchell, the chemiosmotic hypothesis. And it says that basically cells gain energy from redox reactions where you have an environmentally available donor that becomes oxidized. The electrons are transferred to an environmentally available acceptor. And in the process of this redox reaction, ions are pumped from the inside of the cell to the outside. And when they re-enter the cell, they do so through coupling factors. These were hypothetical factors in Peter Mitchell's day. Today we know they're rotor-stator ATPases, and that this is the process that generates ATP that drives forward all other processes. And when he first proposed this, people thought he was crazy, and today we know that this is actually the universal process of generating ATP, and the ATPase is amazingly just as conserved as the genetic code itself, meaning that it was present in LUCA. So when I am thinking about the origin of life, excuse me, I want to get from rocks and water on the early Earth to something like this in about a half an hour. Okay? That's what the objective of today's talk is, and my hope is that when we're done, even if it's not true, that it's better imaginable. All right, so this looks familiar, right? This is the Pope's clothing, right? This is John Barris, and John is the first guy to talk about hydrothermal vents and the origin of life. Many of you have heard of, prebiotic soup and organic soup and the Miller-Urey experiment. There's a different class of theories that work on these hydrothermal vents. John Barris is a microbiologist at the University of Washington, and He actually runs the Pontifical Academy of Science astrobiology program, or did for some time. And these are actually the Pope's real hat and real things. So he, from the where they were, he went ahead and put them on. So John Barris is a very interesting guy, and these ideas go back to the 1980s. Okay, we need to say that. And also, Mike Russell and Deb Kelly, I'll be leaning on their work a lot as well. Mike came up with a theory in the mid-1990s. He says, we propose that life emerged from growing aggregates of iron sulfide bubbles containing alkaline and highly reduced hydrothermal solution. These bubbles were inflated hydrostatically at a sulfidic submarine hot spring sited some distance from an oceanic spreading center 4 billion years ago. What is he saying there? Well, he was talking about a kind of hydrothermal vent that Deb Kelly subsequently discovered. She's also working at the University of Washington. And she discovered a kind of vent called Lost City that's not the black smoker type. The black smokers are much too hot for life. This is, it contains roughly 70-degree hot water with very rich in hydrogen. It contains sulfide, and it contains bubble-like microcompartments. So what are we talking about here? This is a rock that Mike discovered at the silver mines in Ireland, and it's called a botryoid. So geologists know this kind of rock. It's basically little bubble-like things here that you can see. It's a fossilized hydrothermal vent. He's looking at a hydrothermal vent where water was coming up out of the earth, in this case, 350 million years ago. The water was iron-rich, and it was interfacing with sulfide. This is a little chimney here. It's about 1 centimeter in diameter, about as big as this. And where the iron meets the sulfide, you get precipitates of transition metal sulfides here that make these bubbles that are inflated because of the water coming up from the bowels of the earth. And that's why they make these microcompartments, bubble-like microcompartments that are inflated hydrostatically. Excuse me. So these compartments are naturally forming inorganic microcompartments that will play a very important role in today's talk. Can I try this instead? My arm is going to get tired. Can I turn this on? Does that work? Okay. Yeah, I'm going to get I'm a little bit sick, so I'm going to get tired holding that. All right. So, is that okay? Yeah. Okay. So these compartments will play a very important role in today's talk for two reasons. Number one, they will be providing compartmentation. All right? So compartmentation is important. Regardless of what chemistry we assume at the origin of life, the chemicals that are synthesized have to be concentrated around their site of synthesis, because if they diffuse off into the ocean, then they're useless and will never be available for the next reaction steps to get to higher complexity. Okay, one solution around this is say, well, what if we just assume a little warm little pond where they can accumulate and then dry out and stuff like that? Well, the problem with warm little ponds is, number one, on the early Earth, the oceans were twice as deep as they are today, because the water that is today bound in the crust and the mantle was in the oceans. There was no land. And number two, even if there was land, it better not rain, okay, to wash it out. So these compartments here serve as naturally forming microcompartments that can concentrate the products around the site of synthesis. And the other advantage that they have is that they are catalytic. They are made out of transition metal sulfides. Now, many of you may or may not know that there's a field of chemistry called organometallic chemistry, and it uses metals as catalysts in industry. And just to briefly explain, to recapitulate, this is a metal ion, and the reason that the transition metals are catalytic is because they have large unfilled d and f electron shells that can hybridize and form metastable bonds with carbon and nitrogen to make those atoms more reactive so that they can react out here, and then when the reaction is done, the D and F electrons can go back to their standard orbital in the transition metal, and the reaction product leaves the atom unchanged. Okay, so what organometallic chemistry does is coordinate those orbitals in such a way that they can actually modulate the reactivity of the active orbitals, and but the property of catalysis is a natural property of the elements themselves. So if we're talking about hydrothermal vents and iron sulfide rich minerals, it's not the sulfur, it's the transition metals that are doing the catalysis. So we have naturally forming microcompartments with catalytically lined walls. That is a good combination, I think. Lost City, excuse me, I'm so sorry. Lost City is not made of iron sulfide. It's made out of calcium magnesium carbonate, but nonetheless it makes these spires that are up to 60 meters high, and if you cut them open, here's 500 micrometers, or here's a millimeter, that's a millimeter in scale. They are full of these naturally forming microcompartments because the precipitates are formed at the interface, and so you can imagine that we have this large system of intricate microcompartments where some kind of reaction might take place. We don't have any 4 billion year old hydrothermal vents fossilized that we can look at, but we can imagine them having existed on the early Earth because the chemistry that gives rise to the convection currents that generates these hydrothermal vents has been the same on the Earth for 4 billion years. Okay, so given that, this is basically the model. Mike Russell and I, together in a paper that appeared in the Philosophical Transactions of the Royal Society of London in 2003, it goes like this, right? We start out with hydrogen and CO2 as the redox couple. This is the electron donor. This is the electron acceptor from our Peter Mitchell figure at the beginning. And that is the redox couple that's going to generate the energy that we need to produce reduced carbon compounds. It's also very important to note that in the interaction of hydrogen with CO2, the equilibrium lies on the side of acetate and methane. Okay, and that's why methanogens and acetogens can glean their energy and live from reducing CO2 with electrons from hydrogen. We have these imaginary iron-sulfur compartments that are a labyrinth, sort of like a honeycomb of territories where molecules can react, promoted by the activity of the transition metals, giving us things like thioesters or amino acids, sugars and bases, simple compounds, organic precursors that are concentrated around their site of synthesis at a hydrothermal vent, and can become more complex, allowing us to reach higher levels of complexity. And what's also interesting, there's a difference here to the black smoker types of vents. The black smokers sit directly on the magma spreading zones, directly above the magma chamber, and they have a very short life span on the order of decades. Lost City is 15 kilometers away from the spreading zone, and therefore it's just sitting on the ocean floor. It's fed by a completely different convective current and has been active for about 100,000 years. So we can imagine this kind of time that would allow us to get higher reactivity. Now, when this paper appeared, there was a lot of press, and one day the Catholic Academy, about 3 weeks after the paper appeared, the telephone rang in my office, and I went to the telephone, Martin, and I said, "Hello, Martin. Yeah, Martin, this is Schmidt from the Catholic Academy in Stuttgart." Yes. We'd like to talk to you about our paper, about your paper. Did I do something wrong? No, we'd just like to talk to you. Okay? I said, okay. And I said, so what we'd like you to do is come down and give us what's called an impulse. It's a 2-hour lecture where they're allowed to ask as many questions as they want and just inform themselves about what's going on. Okay? And I very much enjoy interactions with the church. I've also given this talk in the Vatican. The Pope wasn't there, but we didn't call it Origin of Life. We called it Early Evolution. Because that's Origin of Life is something different. And so at any rate, I was giving this talk, and there were only 12 people in the audience there in Stuttgart, a very suspicious number. There were 3 priests, 3 scientific theologians, 3 philosophers who were asking me to define everything. Could you define life? Could you define a gene? That's actually very annoying because biologists can talk about these things without having to define it. And 3 physicists. Can you ask me why they always get the physicists involved in origin of life problems? I mean, it's a biological problem, right? But it was 3 physicists. At any rate, about halfway through the talk, I was talking about, amino acids and bases, and one of the priests interrupted me. He says, Dr. Martin, I can see how when you get to these simple organic compounds here from the very simple compounds here, inorganic, that's okay. That transition is plausible. And if you have organic compounds, the complexity of a vitamin or so, like cobalamin or pyridoxal phosphate or thiamine, then I can see how we might get to higher complexity in something like an RNA world. But that transition here, from the simple molecules to the complex molecules, wouldn't that work better if you used a little bit of God? And the answer is, oh yes. Excuse me, oh yes. And the more God you use, the better it works, okay? If we use a whole lot of God, the theory works really, really well, but we can't do that, okay? As a scientist, we can't, when we don't know, we can't say, okay, it was a miracle, although that's the easy way to do it. We just say we don't know, okay, and this is the way that science works. We don't know, and then as new things come along that we understand, and we understand things better, the facts change over time. I also like to say there are no facts, there are only observations and their interpretation. All right, so there are, one of the things that's very important about this theory is that these transition metal sulfides, they make clusters that are not different from the iron-sulfur clusters in modern proteins, whether it's whether it's 2 iron, 2 sulfur, 4 iron, 4 sulfur, with or without nickel. And so what we're saying is that these iron-sulfur clusters in modern proteins that perform electron transfer, redox reactions, and also catalysis, we'll show an example later, are not inventions of the biological world, but it's just that when we got to genes and proteins, the first pre-life forms learned to incorporate these catalysts into proteins to make them portable and specifiable with genes. And so that is life learning to modulate this catalysis a little bit to introduce more specificity into the reactions. The other part of the theory that is that there was no free-living universal common ancestor. Rather, the universal common ancestor was a captive within the system of inorganic microcompartments within which it arose, and that the archaea and the bacteria independently learned to escape from this vent because the constituents of their lipid membrane, the isoprene ethers of the archaea, have different stereochemistry at glycerol phosphate than the fatty acid esters of the bacteria. And furthermore, the peptidoglycan of the bacterial cell wall has nothing in common with the S layer of the archaeal cell wall. And for any other pathway, we would say those were independent inventions. And that's what we are also saying here, is that actually this is a fairly late, this compartmentation from the environment specified by genes is a fairly late invention. And let's remember that in order to get to something that's a free-living cell, in order to put things in context, we have to have like 1,000 genes or 1,500 genes. It's a very, very complex process here, and this is just a rough sketch of how it might have been possible in terms of carbon and energy. So where are we getting the carbon and energy from? This hydrogen, where does that come from? And this CO2, where does that come from? All right, well, where the hydrogen comes from, we don't have to make it up. It really is there. It comes from a process called serpentinization. And the serpentinization process is basically this. This is it's known by the geochemists for many years. They're beginning to understand it in a little bit more detail. This is an equation written by Wolfgang Bach, and basically it goes like this. All right, we have the Earth, we have the submarine crust with these rocks. Okay, this is magma that's cooled. It comes up at spreading zones. It's iron magnesium silicates, and that interacts with water. And this is iron 2, right? Iron magnesium silicates. This is iron 2. And a little bit of that iron 2 is oxidized to give you iron 3, which ends up in magnetite over here. And the electrons that come off the iron 2 end up on water to reduce water to molecular hydrogen. And the oxygen water ends up also in magnetite over here. You can see from the oxidation state of iron over here, there's some iron 2 and some iron 3 in it. And that hydrogen comes up from hydrothermal vents. Lost City generates about 10 millimolar hydrogen all day, every day. Some vents have up to 50 millimolar hydrogen. And some vents generate a mineral called awaruite that has iron nickel in it, metal, pure metal, and that means that there's up to 200 millimolar hydrogen had to be present in some of these hydrothermal vents during their genesis in order to generate that metal. Okay, so that's where the hydrogen come from. Furthermore, the process is energy releasing, it's exergonic, and it has been going on since there is water on Earth because you can't stop it. All right, so that's where the hydrogen is coming from. And this is why this theory is working with the laws of thermodynamics, because in the equilibrium, in the reaction of hydrogen with CO2, the equilibrium lies on the side of acetate and methane, and that's why acetogens are able to generate energy from this reaction here. And it's in the process of generating that acetate, they have a free energy change of minus 104 kilojoules per mole. They use that to pump ions to generate their ATP, and methanogens go a little bit further. They reduce the methyl group here all the way to methane. That's why this is a slightly more exergonic. And both of them satisfy their CO2 needs also with hydrogen and CO2 using a pathway called the acetyl-CoA pathway, which is a linear pathway of CO2 reduction. And it is also exergonic even to the level of the energy-rich thioester. Okay, so some people say that organisms that use this pathway are getting a free lunch that they are paid to eat. It's clear that we're suggesting that acetogens and methanogens are ancient, ancestral, and that they furthermore have preserved traces of the most ancestral pathway, which we're looking at right here. All right, this is just a reminder that people like Volker Müller and Rolf Tauer and Steve Ragsdale and Georg Fuchs, bacterial physiologists and archaeophysiologists, have done a lot of work on these pathways, and they can generate overall schemes for the reaction. And what it is hydrogen plus CO2 gives you acetate in the case of the acetogens. That generates an ion gradient because that's coupled to pumping, and sodium ions come back through the cell and they generate their ATP. So they have to use chemiosmosis. For methanogens, it's also a very similar process. It's just that the pumping is fundamentally different. They don't use this protein called RNF. Rather, they use an energy-conserving hydrogenase, but they also have to pump ions in order to harness that chemiosmotic gradient. The main thing that I want to convey here, though, is that acetogens, for every molecule of CO2 that is fixed to cell mass, 24 molecules of CO2 leave the cell as acetate. For the methanogens, it's about 20 molecules of CO2 that leave the cell as methane per CO2 fixed. All right, it's about 1 to 20 the amount of carbon going through the cell versus the amount of carbon that's fixed as the cell. Life is a side product of the main energy releasing reaction. And that's true for all forms of life, right? The main energy-releasing reaction generates the energy. We generate, what, a body weight of ATP every day. Roughly a body weight, about 60 kilograms of ATP every day. So life is an energy-releasing reaction. I won't go into lots of details. All we'll say is that this acetyl-CoA pathway consists of making a methyl group. Okay, the proteins involved in this methyl synthesis are different in the acetogens and methanogens. And you see lots of cofactors here. Okay, what we're saying is that actually the methyl group was there first, and that when cells were, and this was provided by geochemistry, and then when cells and genes came along, they learned how to synthesize that methyl group with a chemistry that's specified by genes. That may sound like putting the cart before the horse, but actually all we're saying is that the chemistry is conserved of the methyl group synthesis. The genes are different, and all that means is that genes do not perform feats of magic. Enzymes do not perform feats of magic. They just catalyze reactions that tend to occur anyway. And there's new interesting work by a guy named Joseph Moran showing that actually the synthesis of these methyl groups occurs spontaneously under simple laboratory conditions. All right, if we have that methyl group in the chemistry of the acetyl-CoA pathway, that then reaches an enzyme called acetyl-CoA synthase, and its mechanism is very simple. You start with CO2 down here. That generates a molecule of carbon monoxide, and that is then, that binds a nickel group. The methyl group from the methyl synthesis branch binds the same nickel, carbonyl insertion. That nickel-bound acetyl group is cleaved via thiolysis to give us acetyl-CoA. That's how the pathway works. But the important thing here is that it's transition metal sulfides that do all the catalysis, and that indicates that this pathway is probably very ancient. I'm not going to get into that. I will get into that, though, and just say that what's interesting is that the catalysis is actually performed by nickel zero here. If we look at the carbon monoxide synthesis step, there's a 2-electron reaction from nickel 2 to nickel 0. Okay, ferredoxin has to reduce that nickel, and then it binds the CO2. The reduction of CO2 is always a 2-electron reaction in metabolism, and that points to a very important role for these metals. And I'd just like to point out that we've been working on this with a PhD student. Also, Joseph Moran has been working on this. And there's a particular metal called awaruite. It's an iron nickel. It's pure metal, okay? It's not a silicate or anything. It's just a piece of metal, raw metal, iron and nickel zero. And we have been able to show that if you react CO2 with this metal at 200 degrees, we can get CO. And if we react it with carbonate and hydrogen, we can get methane. And Joseph Moran has been able to show that if you react it together with Joseph, he's been able to get acetate and pyruvate. And with CO2 and awaruite and water, we're able to get formate, acetate, and pyruvate in very good concentrations. And that's actually very interesting, because what it indicates is that the intermediates and end products of the acetyl-CoA pathway arise spontaneously in the interaction of CO2 with compounds that are formed in hydrothermal vents. I think that's very significant. I think that's very significant because we've not had that before. This is brand new. Joseph's paper has just been accepted. He scooped us on this. We were working on it. And it's very interesting to note the reason that he scooped us and the reason that we did not see these soluble compounds is because you've got the metals, and the carbon reduction occurs on the metals. So you get a carbonyl group, a methyl group, the insertion occurs on, and the acetyl group remains on the metal, and then another carbonyl insertion to give you pyruvyl, and we didn't see any products in solution. Joseph did this smart step. You have to cleave that off with potassium hydroxide to get it in solution. So we were throwing it away. Well, What did luck falls into the prepared lap? We were not prepared. So Joseph was prepared. At any rate, so that's very interesting to see that the most central compounds in all of carbon metabolism arise spontaneously from CO2. It doesn't even involve hydrogen. It's the metals, and probably the hydrogen is just a means to generate the metals. All right, so that's that. Okay, so what we're saying is that if we have this methyl group and CO2, we can get to, we think, acetyl groups and acetyl thioesters to have carbon and then acetyl phosphate via phosphorylasis all day long. It's also interesting to note that the acetogens use substrate level phosphorylation to generate acetyl phosphate, which we think is a precursor to ATP because it's simpler and it has a higher free energy of hydrolysis, minus 43 kilojoules per mole versus ATP at minus 32. That would give us phosphorylating potential. I think that this could get us in principle to genes and proteins as a source of carbon and energy. Leaving out a lot of details here, okay? I know that. And if we get to genes and proteins, it means we have a ribosome when we have the code. One of the most ancient proteins is going to have to be this ATPase because it's universally conserved. It's present in all genomes. Everybody uses it. The archaea and the bacteria have two different versions. Nobody has... People complain, say, Dr. Martin, how can you possibly suggest that anything as complex as a rotor-stator ATPase is ancient? Nobody has a problem with the ribosome, right? The ribosome is ancient. We know that we had genes. In comparison, the ribosome is this big and has 50 proteins. Nobody has a problem with that being ancient. Everybody has a problem with that being. I don't have a problem with that being ancient either. And the interesting thing is that we said alkaline vents. Lost City has a pH of about 9. It's because the alkaline, the serpentinization reaction generates magnesium hydroxide, making this vent fluid alkaline. The ocean, ancient ocean, is neutral or slightly acid. This generates an ion gradient, and I think that this ion gradient would be very interesting as a source of energy for the first cells that possess this ATPase. That would help to explain why it's so conserved. Now, this also would help to explain why the ability to harness these ion gradients is conserved throughout all cells, but the chemistry to generate this ion gradients is very diverse, and it's furthermore not conserved between the acetogens and methanogens, indicating that they were able to use this ancient and that their next step was learning to generate an ion gradient with a chemistry that's specified by genes. If they can do that, then they can escape, and we get something that is very similar to the modern situation here and Peter Mitchell's cell. So that was that in about 45 minutes. I better hurry up because I'm way over time. Can you give me 5 more minutes? Would you like me to just stop? Are you sure? Thank you. That was a suggestive question. That was sort of like, okay, I'll look the other way. Would you like me to stop? Okay. Somebody said yes. This is the title of the paper. This is, okay. Is there any way to test this? We look at genes, we look at genomes. It's comparative. What I do is basically comparative physiology, but it's also very much oriented on genomes, right? Genome analyses. And this paper came out. We did this paper in 2016. It also got a lot of press. It's the physiology and habitat of the last universal common ancestor. And what we did here was actually quite simple. We've been making genes of everything. Okay, so we make clusters of all proteins. So we take 2,000 genomes that contain 6 million proteins. We do an all-by-all blast of that. That gives us a matrix with 36 trillion elements. All right, and then we turn those into clusters so that we sort all the proteins in those genomes into clusters of natural sequence similarity. It helps if you have a very big computer. Thank you, Computer Center, University of Düsseldorf. We have 12 terabytes of shared memory on one machine. That helps. And then from all those proteins, we weren't asking what is universal people who've been addressing the last universal common ancestor have been doing basically this. We look at all genomes and we look for everything that's universally conserved. Well, we know that. It's the ribosome and the ATPase. We were asking not what is conserved, but what is ancient. And for ancient, we say we want the protein to be present in 2 groups of bacteria and 2 groups of archaea, and that these, furthermore, should be monophyletic. Why 2 groups? Because you can have lots of, if we do this experiment with the 6 million proteins, we get 250,000 clusters. 11,000 clusters contain genes that are present in bacteria and archaea. 11,000. Now, they could either be present in LUCA or they could also be the result of lateral gene transfer later. And we know that there's a lot of lateral gene transfer between bacteria and archaea. So how do we sort that out? We say, okay, we ask them to be present in 2 groups, 2 major clades, 2 phylum-level clades. We don't care which 2. And if that's the case and they're still monophyletic, then it's probably not lateral gene transfer. And this is just to show you a little bit of the kind of data that we're looking at. This is 17,000 clusters of proteins that are only present in archaea. And you can see this black line over here at the edge. Those are the genes that are present in all archaea. It's only about 50, and then 30 that are also present in bacteria. People have been making trees of these proteins in the expectation that they would be able to explain the evolution of what's in the rest of the genome. That's not true, and so we've been looking at what's in the rest of the genome directly to see whether it traces to LUCA or not. When we do that, what we found, 11,000 proteins present in bacteria and archaea, 355 present in bacteria and archaea fulfill the 2-2 criterion and monophyly. These are genes that are present in LUCA by phylogenetic criteria, not what is universal, rather what is ancient. Okay, we find the ribosome, we know that. We find translation, we knew that. We find DNA, okay, we knew that. RNA modification, that's kind of interesting, but we also find physiology. We find carbon metabolism, nitrogen metabolism, energy metabolism, and what we find is summarized here, okay? It looks very similar to the figure that I was showing you before because I drew both of them, but this is what we've got, okay? It's got nitrogenase, it's got iron-sulfur clusters. LUCA had acetyl-CoA pathway, okay? This carbon and energy metabolism that I was talking about before, the acyl phosphates. It had subunits of the ATPase, an antiporter here that convert this hydrogen, this proton gradient into a more stable sodium gradient. And in fact, if you look in the acetogens and methanogens, they tend to use sodium ATPases, although the amino acid changes are minor involved there. LUCA lived from gases, nitrogen, hydrogen, CO2, H2S, and carbon monoxide. Okay? It looked very similar to acetogens and methanogens, which furthermore look very similar to the organisms that inhabit hydrothermal vents today. It had lots of cofactors. Cofactors were very important to LUCA's metabolism. This is the last, almost the last thing here. It had modified bases, and what these modified bases indicate to me is that remember the modified bases, the tRNA, mRNA interactions, the wobble hypothesis. They require methylations and sulfur modifications at those bases so that the code will work like the wobble hypothesis predicted. These are chemical modifications that are almost identical to the environment that LUCA inhabited. We think that what this indicates is that the code arose in an environment where there was lots of sulfur flying around and reactive methyl groups flying around. In other words, the code also arose where LUCA was living. Okay, and that these methylations are just imprints of the chemical environment where the code arose. All right. Methanogens and acetogens are furthermore, if you do the reciprocally rooted trees, they come out as the most ancient groups. That's also consistent. Last thing here, my children ask me, what is this good for? Daddy, what is this good for? Well, this is Enceladus. Okay, this is a picture of Saturn, and this is the E ring, and there's a little white dot over here. That's Enceladus. It's a moon of Saturn. And Enceladus is spraying water out into space. It has liquid water because it has a rocky core that's undergoing pressure from Saturn. Okay, it's a moon of Saturn, and there's friction in here that keeps it hot. That generates circulation and heated water that is spraying out at these geysers at the South Pole. The water spraying. Enceladus is only about 500 kilometers in diameter, and these jets go out hundreds of kilometers into space. That's where the water comes from that the E-ring is made of. And last year there was a very nice publication indicating that there's also molecular hydrogen in these vents, and that indicates that there's probably serpentinization process going on Enceladus. And that means, in terms of origin of life, we don't need, part of this theory means that we don't need the sun, okay? Just need rocks, water, and CO2. And furthermore, Frank Posberg from Heidelberg, who gave me these pictures, has found carbon compounds in these plumes. Their nature is still being investigated. They think that there's something like serpentinization and hydrothermal vents going on under the ice and on Enceladus. I could do the movie now, but I'm not going to do it because I've taken too much of your time already, unless you would like to see a 5-minute movie. Okay, you want to see a 5-minute movie? Okay. Does anybody want to say no? You're very kind to me. Thank you. Okay. We'll do a 5-minute movie, and then we're done. Oh, my back. Okay. So, just a second. I've got a problem with my computer on this screen. Okay, it goes like this. Now I'll use this. So on the early Earth, there was no land. There was only water, and 4 billion years ago. And the reason is that there was the oceans were twice as deep as they are today, and that land that there was not a very nice place to be because of the late heavy bombardment. The bottom of the ocean was a much nicer place to be, but it was very dark. You see how black this is? That's why they're called black smokers. And these guys are sitting directly on top of the spreading zones. We'll see that in a minute. And these other kinds of vents off here in the background, Lost City, I think they're more interesting because the black smokers are 400 degrees hot, the water coming out. That's too hot for life. And watch what the animators did here. I said, could you make this thing grow? And they said, sure. And so they took the cursor and watch. They just pull it up and make it grow like that. I said, can you do that better? And they said, yeah, sure. That'll cost $1,000. I said, it's fine. It's fine. Just leave it the way it is. Okay, so here's the black smokers. The spreading zones are where the magma is coming up, okay, and making new crust that moves apart at the spreading zones. Water comes into contact with that magma. 1,200 degrees, comes up superheated at 400 degrees. Lost City, the water is circulating and does not get in contact with magma. Nonetheless, it gets down to about 3 to 5 kilometers to make serpentinite. Okay, the water comes up chemically modified, and we'll see that here in the next picture. All right, we've got sulfur in the crust. Water is being reduced to hydrogen. Hydrogen coming up in large amounts at these hydrothermal vents. Oxygen disappearing in the crust as magnetite. And this goes on even today, and there's nothing special about it. Then at the vent-ocean interface, the sulfur interacting with large amounts of iron-2 and nickel transition metals in the ancient oceans to give us these precipitates that are basically catalytically walled microcompartments that are sort of growing up here in the background that you can see. CO2, hydrogen interfacing here. And the next series that we see is just a cartoon of the acetyl-CoA synthase reaction mechanism. If you're chemically savvy, please be kind, because that is not actually what happens in the chemical mechanism, but it's very similar. Okay, the generation of CO on this background, and if we have methyl groups, as I was saying, we need methyl groups for this to work. They can donate a methyl group to the same nickel carbonyl insertion to make a carbon-carbon bond. Suddenly we've got an acetyl group, and that's looking very biological, and this actually works in the laboratory as well. It's amazing. And then removal of the acetyl group via thiolysis to give us a thioester, and that comes up real big in the picture, and there are lots of them suddenly floating around in the background. It's because we think that acetyl-CoA is the most central compound in metabolism because metabolism arose that way. And then we're almost done. All right, we get these, then we get to amino acids and building blocks, hand waving. I don't have any specifics on that, but here are amino acids and RNA bases. We know that we had to get there. And then also this spontaneous assembly of RNA into long molecules. Nobody has a clue how that happens, okay, but everybody assumes it to have happened, so we also assume it and we put it in here. Furthermore, all of a sudden we've got little peptides here because we have a ribosome in the genetic code. Boy, that was fast. Nobody knows how the genetic code rose, but we know that it arose in the last common ancestor, so we also have it in here as well. Oops, DNA. Yep, DNA was in Luca. And then suddenly we get to, compartmentation with our own cell wall, and they can bubble off, and it's the evolution of free-living prokaryotes for 2 billion years until the origin of eukaryotes, which occurred later. All right, so that was the movie. And all I have to do now is my credits. If I may. Oh, my back. I'm very sorry. I've overstayed my welcome here. Oh, come on, computer. Please. They're very long seconds. Oh, okay. Right. So, who does the work? This is Chuan. Martina is from Taiwan. Martina is from Germany. Maya's from Germany. Felipe is from Portugal. She's moved on to Vienna. Sriram from India. Madeleine also from Germany. Shiju also from India. They've moved on, and if you notice they're all smiling here, it's because, well, you can't really read this very well. We've had a lot of very nice papers recently, and that keeps all the students happy, and it also keeps my grant application people happy, and it keeps me reasonably happy. Although we can't quit, we have to keep on working. That's the problem. When you keep working, you have to keep on working. So that's that, and these are friends and colleagues. These are the team. These are the people who gave me the money, and I'm sorry that it took so long. Thank you very much for listening. I'll be glad to take any and all questions.
So there is some questions in the audience?
Anything, and please be as critical as you want, please.
You define the last common ancestor, but I think that this definition comes from the top, in the sense that we select organisms that are already organized in a certain way. There is DNA, RNA, proteins, and so on. My question is about the fact that if looking at the conditions in which life may be developed, don't you think that that should be more last common ancestor, in the sense that how do we can imagine that in different parts of physiological and physical, whenever you want, of the life on the sea and so on, growth, we got the same mechanism?
Okay. I need, okay. You've got a point, okay. The colleague just won the argument, okay? So could it be that it's actually really a lot more complicated? Could it be that it's a lot more complicated than you're saying? And the answer is it's certainly more complicated. And furthermore, there could have been lots of different things that went on, and all we've got is what survived, okay? at the same time, if we look around among the cells we know today, why do we even, a good question, why do we even believe that there was a universal common ancestor? Well, the code. I think the best evidence that we have that everything traces to a single common ancestor is the genetic code. It's the same everywhere, some minor modifications, but those are exceptions that prove the rule. And the holochirality. Haldane. I was reading Haldane, 1929, the other day, and he was also pointing out that the L amino acids and the D sugars, that this chirality also is evidence. He didn't know anything about the code back then for common ancestry. So I think there's, and unless we say that the code arose by convergence, then that's the best evidence we have. Now, there are also theories that the code arose somewhere and then was transferred around to all these different life forms. It could be, but I think Darwin's principle that the vertical inheritance is probably a better assumption there. And again, there are many things that we can imagine, but I'm trying to draw in this particular effort here, I'm trying to draw the shortest possible connections, at knowing that I'm doing a lot of shortcuts. And you might say, well, what about the synthesis of amino acids? What about the synthesis of bases? And the answer is, we don't have any good answers here from the laboratory that would support that. We've got acetate, methane, formate, pyruvate, stuff like that. Nothing with nitrogen in at the moment, okay? So there are a lot of shortcomings to what I've said today, and I'm not making a wide-ranging claim. It's basically focusing on the carbon and the energy. That's important to me. How we got to RNA molecules and things that are more complicated is a completely open issue. And also, okay, some people get really tight on me and they say, well, could it be, that there were lots of other things that you're only thinking about the things that are alive today. There could have been lots of other things that went extinct. And then I say, yes, that's true, but life is short. And so let's focus on the things that we see, okay? And when we're done with all the things, the evolution of the things that we know, then we can worry about the other ones as well. Okay, so there's a lot of philosophy and a lot of aspects in the question that you asked that would last for a long discussion. Okay. Thanks. I've got a problem with my back.
About 6 months ago, there was an article in Scientific American of colleagues claiming that all this deep springs, is not good. That the idea was that life developed in hot springs at the surface. The argument was, I believe, that concentration came about through temporal drying out into layers instead of in little bubbles. Now, what is your response?
My response is, that's fine. Let me see. It's Dave Deemer and some colleagues, and there's a big debate about this. Now, the idea of warm little ponds, okay, and the surface environment, they're welcome to, like I said, I'm not trying to convince anybody. So I'm happy that they got their nice Scientific American article, but the problem that I see, okay, is that it does not connect to microbial physiology. And when you ask, where does the energy come from in their theory? The energy comes from UV light. Now, I'm a biologist, and I happen to know that there is no form of life that we know that lives from UV light. You have visible light that can be harnessed. That's photosynthesis. That comes after you have respiration, and that probably comes after you have autotrophic growth to make the carbon compounds. So what I find satisfying about the vent theory is that, number one, it works with the same chemical reaction that we see driving some forms of modern metabolism. Okay? When we look to see what is ancient in Luca, we get exactly the same physiology, conditions, anaerobes living from gases. That connection is good. And And so and if we look to see what organisms live at modern hydrothermal vents today, that's really interesting. They live from the same thing, okay? And so what this suggests to me, so I'm gonna sell my story, okay? I'm selling these are my selling points. We have this connection between geochemistry and life, okay, that fits very well. And they have a very nice, pretty picture theory of drying and bases and UV light, but it doesn't connect to life. That's not a problem for them, and that's fine, okay? And so what is also true is that in issues like origin of eukaryotes or origin of life, we just have to live with it. There will never be unanimity of opinion. There will always be, because there are no clear answers. Right? Like I said at the beginning, even if we had a reaction where we got E. coli with the wrong code, we couldn't prove that we arose that way. So there will always be that diversity, and that's good. Let people have their, and furthermore, what are they going to say? Oh, we've been wrong for 50 years? No, science doesn't work that way either. And so what we're doing is trying to make the connections between the geochemistry and the microbiology, and it's what's very interesting. This is also I was telling the students earlier about these chemical reactions involving the metals and the synthesis of formate, acetate, and pyruvate. The connections are getting very stronger, getting stronger rapidly. That doesn't mean that it's true. It just means that we can understand it that way. So I'm very happy that they got their own theory, and that's fine. Okay, but it doesn't connect to life, and that's why you can find my theory in microbiology textbooks. But not theirs, because my theory connects to microbiology. Okay, so it's fine, yeah. And do I wish that I had a Scientific American paper? Yes. I don't have one. In the back. Mentation. Yeah. Okay, these are not, okay. You're paying attention. All right. So, number one, these are gray. The bacterial lipids are blue. The archaeolipids are red. And in the LUCA inference, right, where we look for the 355 genes, we didn't find anything involved in lipid biosynthesis, which is actually consistent with what we're saying. Nonetheless, what are these lipids that are putting here? If you look at organic chemistry, the origin of life reactions where you get CO2 reduced regardless of what's going on, a common theme is the synthesis of tar. Okay, what happens is that if you have reducing conditions, you get hydrophobic compounds. All right, so what we're looking at here are not necessarily phospholipids. The implication here is just a hydrophobic layer. Now, it doesn't have to be a hydrophobic layer with, phospholipids, and the phosphorus, I think, is more here in the acyl phosphates as an energy source than it is in phospholipids. These are just hydrophobic compounds. There is also the circumstance that at Lost City they not only find 1 millimolar methane, but they also find traces of ethane, propane, butane, and pentane. Okay, so there is something like a Fischer-Tropsch synthesis going on down deep. And the reason is that the longer these aliphatic chains get, the more exergonic the reaction is. Now, you've got kinetically controlled reactions and thermodynamically controlled reactions. But what we're saying is that these are hydrophobics. They don't have to be phospholipids, and they do not have to be fatty acids, and they do not have to be sterol derivatives, not sterol derivatives, phytonyl derivatives, as they are in the archaeal ether lipids. So, just hydrophobics will do, and the genetically encoded lipid biosynthesis comes late. Okay, and so Now you can say, well, Dr. Martin, aren't you using a lot of spontaneous synthesis from these geochemical vents? Okay, how far do you want to go with this? All right, we can get serious and say, are you saying that like lipids and amino acids and bases are being synthesized by spontaneous chemistry? Name a theory for the origin of life that does not do the same thing. Now, this is where God really helps, okay? Okay, because all theories for the origin of life are confronted with the same problem. We have to have a whole lot of very complicated chemistry arising spontaneously. And that's really interesting if you think about it. Because, please. Because what that indicates is that something roughly the complexity of a ribosome arose spontaneously. And I don't see any alternative to believing something like that at the moment. I can live with it. And it's just because before we have Darwinian principles, it's thermodynamics. Please.
No, I just want to, since I'm a cell biologist, you said that you are a biologist, so I'm familiar with the cells, not these cells. So for me, phospholipids, and he raised an important question, because phospholipids play an important role because it is believed that we separate the cell, the internal part of the cell, from the environment, and this raises an important question, because at a certain point. So first of all, let me tell you. So do you believe that life, thinking about cells, is the result of the chemical reaction, or maybe chemical physical reaction, that give rise to something that is which has properties that are different from the single elements that reacted together? And I like very much your idea, because you said that you show these metals that are very reactive, and when we talk about cells, we talk about the independent irritability of the cells, because they are this capability to respond to the environment, and then to modify them on the surface. Okay, so maybe we can talk later if you. So it's nice, because maybe I can see an independent irritability of the metals or something, so that this guy spontaneously, react.
You've touched on a lot of really interesting thoughts there. The sum is more than the individual parts. Okay. This is also one of the problems with organic soup. We've got old organic soup theories. And, I mean, we can do the experiment. We can get a can of chicken soup, a can of goulash soup, or we can take 3 frogs and 4 carrots, homogenize them. We have all the molecules that we need for life. Put that sterile in a can, water as a control, at 4 billion years, and just wait for new life to evolve. And it won't evolve because life is not just a process of reorganizing pre-existing components. The sum is more than the cell is more than the sum of its parts. That is, if you decompose the cell, you can't put it back together again.
Exactly.
And what's also really interesting, if you think about the fact that we've got this chemical reaction, right, that gives rise to life. It's one and the same chemical reaction that has been going on in uninterrupted continuity since the origin of life, regardless of how it arose. It's that chemical reaction that's giving rise to the side product, not us. So independent irritability. It's also kind of interesting to think this is sensing the environment. Okay. Now, I think the sensing part is actually advanced, because if we look at highly specialized cells like acetogens and methanogens, you give them nitrogen, gas, hydrogen, CO2, H2S, a few mineral salts, and they grow. The specialists that are specialized to growth like that, they're called hydrogenotrophic, and microbiologists say the state of gene regulation is on. They are just on, because you give them their reactants, and they do a chemical reaction to make more cells with a doubling time of about 30 minutes. And and they have no decisions to make because they know exactly one thing to do. That's also the things that live in the crust today. It's really interesting. About half the biomass on Earth lives below the crust. So it's a big biomass down there. And so the decision-making process, do I have the environment where I want to start expressing gene, means that we've already reached a more differentiated state, and there are different ways to make a living. That gets into gene regulation, requiring responses and sensing a more advanced state. That's really interesting.
No? Bill, thank you very much for your seminar and
Okay.
for coming.
Thank you. Thank you.